have a slide up. Okay, next up we have Matteo Carandini, who's an old friend and colleague, who I believe holds the record for simultaneous recording from the largest number of neurons all at once, at least in a vertebrate. Is that correct?
I have no idea.
Yeah, okay. Anyway, he's going to talk about testing the textbook model of brain function.
Yeah, thank you so much for having me. I'm nervous because this is my alma mater, and somehow when you give a talk at your alma mater, you really don't want to screw up. So I'm going to do my best not to screw up. And in particular, I think I will not screw up because I have really wonderful data to show you. These data come from a postdoc called Nick Steinmetz, and his name will be down here very often. None of what I'll show you is published. I also need to thank Daniel Wolpert for introducing Candel and Schwartz's book, Principles of Neuroscience, which will be featuring prominently in my talk. So it would be great. I mean, we're in an era of fantastic collaboration between neuroscience and machine learning, and the areas where the two are meeting are, in deep networks, which have a lot to do with how the visual system works in the first 150 milliseconds, as you'll hear later in this symposium, and also reinforcement learning, which comes from behavioral work, but also brain work. It's been a fantastic loop between theory and experiments. But wouldn't it be amazing if we could go to Leslie and say, Leslie, why do you have your robots work that way? Why don't you do it the way the brain does it? And then she would say, well, how does the brain does it? And the answer is we have no clue, okay, really. We know a lot about bits and pieces, but there isn't a textbook that says, oh, okay, by the way, this is how you go and grab a cup. And this is how you make a visual decision and move your muscles. And then you can read that book and say to your roboticist friends, hey, just read the book, okay? We don't have it. So, but let's see what the current book says. Now, here's an extremely simplified view of what the book says. Taken from the standpoint of a visual neuroscientist like me, where I like to think of sensory inputs coming in and motor actions coming out. And I'm not saying that this is specifically written in this book, but this is the gist of any textbook I could find about how the brain works. It will say that there's some sensory processing, some more sensory processing, maybe there's something called decision making, even not everybody agrees on that. Then there's action selection and execution, and then motor output. Of course, this doesn't operate in a vacuum. There's also an outcome of those motor outcomes of output. You're going to get some reward or punishment or something. You're going to process it, and then you're going to learn somewhere. Okay? And apologies, I know many of you have better views of how the brain works. I'm just saying they haven't trickled into the textbooks yet, I think. And you might think this is super naive, but guess what? This is actually also how a deep network works, really. There's some sensory input, there's a bunch of stuff happening, filtering and more filtering and thresholding and more filtering and thresholding, and then there's some outputs, and then some outcomes and learning comes back through. So that's still the basic idea. Now, is this how the brain works? One way to figure out if that's how the brain works is to train, for example, a monkey to do a sensory task and go record all over the brain and figure out whether the signals follow this kind of sequence. And this was the heroic work done for example, in the Romo lab. This paper here summarizes, I don't know how many years of work by how many people. And the task here, this is a macaque cortex. The task involved a monkey feeling some fluctuation on the finger, and therefore S1 is a primary sensory area for this task. S2 is more sensory processing, and all this stuff up here would have to be places where you make a decision or action selection, assuming you're thinking that this whole thing happens in cortex. That line of work was tremendously successful. I just want to point out one limitation of it, which is that it didn't really tell us about an exact flow of how things would flow through, the time from stimulus onset and where you can find some signals that you can call decision. Seems to be pretty homogeneous around here. But I don't want to criticize this work, but I just want to tell you that this is, I think, the way to go, actually, to answer this question. And in the lab, and in many labs, we're now trying to answer it in a species that is a bit more distant, but friendlier, in my opinion, the mouse. And to see how this flow of activity during decision making flows through the cortex, we train mice to perform a task that is a simple visual detection task. Here is shown as a detection task. Here you might see two stimuli sometimes, and the mouse needs to decide which one has higher contrast. We run it both ways. Let me first have you concentrate on this. The mouse is head fixed. It drinks when it gets a reward. It's moving a wheel that you can't really see because it's too dark, but here's the wheel. It's a Lego wheel as it happens. This is the pupil, in case you want to know how aroused this mouse is. And this is the stimulus that the mouse is seeing actually around it's on 3 screens. And when the mouse sees a stimulus of higher contrast, it needs to move it after a certain delay to the center of the screen. And mice get really, really, really good at this task. So when there's a lot of contrast on the right side, they always go on the right side. When there's a lot of contrast on the left side, they always go on the left side. When there's no contrast, we train them that the correct thing to do is to hold the wheel. And this will be very useful when you want to interpret results of manipulations, of brain manipulations. And the mice pretty much get it. You can see here at 0 contrast, 50% of the time the mouse held the wheel. Other times, the mouse decided for left or right, okay, because there's uncertainty, as you heard from Daniel Wolpert. Okay, so now that you know this task, the question is, where in the cortex do we see signals related to this task? Will we see responses in V1, for example, or in what I'm going to call V2 improperly, because the mouse has some beautiful visual areas, secondary visual areas. I'm going to put it in quotes every time I remember. Will we see signals in secondary motor cortex and then maybe movement execution signals in primary motor cortex? The first thing we can do is use wide field imaging of calcium indicators, because you can actually literally buy mice that shine in the dark, and where all you need to do is make the skull transparent and see activity. And this is real-time activity while this mouse is doing this task. This is what's happening in the cortex. And I'm not going to ask you to interpret it. I'm going to show you some interpretations of it. But as you probably will hear from Anne Churchland, I don't know where she is, if she's here. Anyway, as you probably will hear from her, a lot of this activity has to do not with the task, but with whiskers moving, body movements, all sorts of things that are not directly related to the task. But what I want to focus on is what happens at the times that are absolutely tied to the task. This is the activity of the brain, of the cortex. Sometimes I say brain because I'm a cortex-centric, I don't know. The problem with the cortex is you always think it's the best place on earth. But it's not, as you will see. This is what the activity of the cortex at the time the stimulus is shown. About 100 milliseconds later, there's clear activity in the part of primary visual cortex that is processing the stimulus. There's a stimulus on the left side of the visual screen, so the right side of the cortex is active. There's activity in the secondary areas of the visual cortex. This is also called parietal cortex, by the way, up here. And there's also activity here in secondary motor cortex. 250 milliseconds later, all hell breaks loose, okay? Massive amounts of activity in primary sensory cortex and in primary motor cortex, okay? Let's see. I want to tell you one more thing about this activity up here and down here, okay? Which is that the activity seems to follow a flow. And let me show it to you here. We're going to choose a region of interest in primary visual cortex, one in secondary visual cortex, one in primary motor cortex, one in secondary motor cortex. And you can see that the activity follows this regular flow from V1 to V2 in quotes to M2 to M1, which is actually the textbook view, okay? So let's be happy about the textbook, at least for this slide, all right? We're going to get sensory input, some sensory processing, more sensory processing, some kind of decision making maybe, and then a motor output. So as I said, I just went ahead of myself. This is pretty much the textbook view. Now, let's ask, though, these activations that I showed you, are they needed? Are they causally related to what the mouse is doing? And so let's check for causality. And what we're going to do now is use a trick that was invented by Massimo Scanziani and then kind of expanded on by Karol Swoboda. And the trick is you get a mouse excuse me, that has channel rhodopsin in inhibitory neurons, so that when you shine blue light on those that part of the brain, the inhibitory neurons fire like maniacs, thus shutting down the excitatory neurons. And that's an invention by the Scanziani lab. Karol Swoboda added the fact that you can scan through the cortex to inactivate wherever you want. So we're going to do this game of inactivation, and now I'm just going to show you the result of inactivating this region, which is primary visual cortex and secondary visual cortex. We have not found much difference between these points, so let's pool them here. When we inactivate this, as you might expect, we severely, oh, by the way, this inactivation is done throughout the presentation of the stimulus, okay? We severely reduce the ability of the mouse to see stimuli on the left, and we do nothing to the ability of the mouse to see stimuli on the right, which makes sense, right? Because the right visual cortex has been inactivated, and so it has a problem seeing these stimuli on the left. And so let's look at what happens. For example, take this stimulus that has 50% contrast. It's extremely visible normally, okay? The mouse is perfect at it usually. Sorry, here, it's perfect at it. It always goes left. Now, when we inactivate this chunk of cortex, the mouse makes a bunch of mistakes. What kind of mistakes? Well, sometimes it chooses no-goes, meaning it really thinks there's nothing on the screen. And sometimes it thinks maybe there's something on the right, because maybe there's more activity here than there in those trials, okay? So this works, and we can now build a map and ask what happens when I inactivate all of these regions. And now the circle size is going to change meaning, and instead of meaning the size of the inactivated region, it's going to mean how big an effect we saw, okay? So this is a summary of the results. It is actually a summary of the result that we obtained when the mouse was seeing two identical contrasts and had to guess to go left or right. And so you see that when you inactivate, and by right here I mean rightward, not right as in correct. And as you can see, you have effects that are symmetrical in visual cortex, and you also have effects in secondary motor cortex. And if you now modify the experiment slightly, so that instead of inactivating throughout the whole presentation of the stimulus, you inactivate only for 25 milliseconds just before the stimulus arrives, or at the time of the stimulus, or a little later, you can actually distinguish fundamentally different timings at which these regions are used. And here's the result. If you do a 25 millisecond pulse in which you shut down V1, you basically impair, and here we're plotting reaction time as a way. This is the normal reaction time of the mouse, and this is how we lengthen the reaction time, so, essentially interfering with the mouse's ability to do the task when we do this manipulation. But by the way, the mouse will end up doing the right thing anyway, because we're only interfering with 25 milliseconds, and the stimulus is on for a longer time. Okay, so the only thing we can do is really delay it. And as you can see, if you inactivate V1 near the time of the stimulus, you really interfere with the animal's ability to do the task. But for M2, that comes later. Again, consistent with this idea that this region is needed before this region. Okay. So now I want to point out, though, this massive hole here. I had shown you, using wide field imaging, that there's a massive chunk of activity in primary motor cortex and primary sensory cortex, and those regions which are now plotted here are not needed for the task. Okay, so beware of correlation and jumping to conclusions, such like you see a lot of activity and you think it could be related to the task. And so now we clearly need to go down into the brain to figure out what's necessary for the task and what's not. And also we would like to get out of this system of looking at huge chunks of brain. By the way, this is probably 1 millimeter if you want to get an idea of the size here. I would like to go down to the individual neurons, and to do that, and study regions that are below the cortex. And to do that, we're going to use this technology called NeuroPixels probes, and this is published, and it's a result of a fantastic collaboration between various funders, scientists, engineers. It's really an exciting moment for neuroscience. You might have heard about them. These probes are 1 centimeter long, which is actually too long for a mouse brain. The mouse brain is not that long. But from these, you get to select 384 of these channels, and what comes out of the probe is already amplified and digitized. So you don't need anything else in the lab. It could, in principle, go straight into a laptop, except that it doesn't speak FireWire or USB, so you actually need a converter box. But other than that, it could go into a laptop. With this kind of technology, and just to show you where this technology is relative to the past, this is a little plot. This is shown. It's in this review paper. It's a little plot that shows the number of recording sites per shank as a function of year, and neuropixels is way up there. I'm afraid it's going to contribute to the number of pages of Kandel and Schwartz, but what can I do? And so it's a really wonderful technology. With this technology, you can stick, for now, we stick 2 of these at once in a mouse while the mouse is doing the task. And here are recordings. You know, I've skipped the fact that there's spike sorting and all sorts of interesting processing going on. Here's a result of measuring at the same time while the mouse is doing the task. This was an older neuropixels. It looks clunkier than what it actually is currently. And here are recordings from something fairly anterior in the brain, this section, M2, anterior cingulate, limbic cortex. These are recordings from V1. By the way, notice that V1 is doing all sorts of stuff when there's nothing on the screen, which is funny. And now we know that it has to do with all sorts of things that the face is doing, etc. And the dorsosubbiculum and thalamus, and so on. And now, if you stick electrodes all over, one of the big problems you have is to reconstruct where were they, because you can't do lesions with these electrodes. And so we found a trick, which we've copied from the Allen Institute, where they invented it, which is to dip the electrode into a relative of dye eye, which is red, and then clear the brain and make it transparent, and then put it in a very simple but complicated sounding machine called an optical projection tomography machine. And so the result is you use the autofluorescence of the brain to find the structures. For some reason, this movie refuses to loop. You know what? Okay, let's go back, see what happens that way. Forward. I don't know. Anyway, it's a nice movie, but my computer refuses to show it. Anyway, you would have seen, maybe if you were lucky, 2 red traces, because this is coronal sections, and you could see the electrodes. And here you would have seen 2 red dots following the electrodes. Anyway, sorry, I'll put it up later when we're done with the talk. And now, with this method, we can align the recordings to the Allen Common Coordinate Framework, which is the average shape of a mouse brain, annotated down to 10 microns. We can now record from, we've recorded from 5 mice. In each mouse, we've put electrodes 2 at a time. And so now we have data from 20,000 neurons or more, recorded in more than 40 brain regions, and in 5 different mice. Now, to tell you our results, I could, there's a lot that you can do with these data, okay? I'm going to do the simplest kind of thing. I'm going to tell you averages of firing rates in different brain regions. No fancy analysis. We do fit GLM models, etc. I won't tell you about that. I will tell you nothing about correlations between neurons. So in principle, yes, we could have recorded these neurons 1 at a time. I think the noise would have been amazing, but there's much more to do. But even then, there's already a lot of interesting results. So first of all, let's do, I think there'll be 1 model, actually. Let's first look at the activity where, as it progresses from the time we turn the stimulus on while the mouse, or this average mouse, is doing the task. The first place where we see activity in the cortex is V1. Then we see it in V2, moves on to M2. This is with electrodes, okay? These are the number of neurons that have been recorded in these places. And then it moves to M1, and you'd say, you just rediscovered what you had gotten with the wide field imaging. But let's go now deeper in the brain. Oops, superior colliculus, responding way before V1. I have to put a caveat, which is that we do have recordings from the lateral genicular nucleus, which presumably will also respond before V1, but we do not have electrodes in the place that is looking at the stimulus, and therefore I'm not showing that here. It was bad luck. We need to keep on going until we hit the place that is looking at the stimulus. So don't assume the superior colliculus is so magical. I'm sure a lateral genicular nucleus also would have responded early, and the retina even earlier, right? Okay, let's progress. Now we are in the caudoputamen, which actually responds before M2. Okay, and now we're in the midbrain reticular nucleus down here under the superior colliculus. This is an awesome place, by the way. Holy moly, you'll see later what it does. If I were to start again, forget about cerebral cortex. Midbrain reticular nucleus, this is where it's at. Okay, and so you'll think, yeah, it's not doing anything that interesting, but this is synced to the stimulus. Wait until we sync things to motor behavior. Okay, okay. So now I'm going to essentially plot the same data, but instead of showing you only 1, 2, 3, 4, 5, 6, 7 brain regions, I'm going to show you a ton of brain regions. Okay, and here they are. Here you can see. You know, it's hard. I don't even personally know all of these acronyms. I'm bad at acronyms. But essentially, we have superficial, and now activity is a heat map. You can see that a ton of areas have been activated since the time from stimulus. We have the superficial layers of the superior colliculus, primary visual cortex, then one secondary visual cortex starts to show up, then LP, which is down here, and it's part of the thalamus. I bet LGN would have been earlier, sorry. It comes up down here for stupid reasons. Then there's more secondary visual areas, and then we start getting more frontal areas of the cortex. We're in the midbrain, and I'm going to spare you the details. Now, you might say, whoa, all of these are visual regions? That's wrong. How can it be? The textbook says the vision should be in only a few of these places. And the textbook is correct in that sense, because if we now play back the visual stimuli to these mice, while the mice are not engaged in a visual in a task, we find activity in very few places, which are the places where the textbook says the activity should be, okay? And similarly, if you know, there's a beep that tells the mouse that it's okay to turn the wheel, we can play that beep and ask which of these regions are active, and only very few regions are active. And again, those are the ones that the textbook would tell you. Incredibly inferior colliculus. I don't know if it's here, by the way. Stunning auditory place. Okay, but now I want to give you what's surprising. And what's really surprising is that these regions that I've now, you know, I'm going back. I've shown you this aligned by time of stimulus onset, time of stimulus onset of a passive visual stimulus, time of stimulus onset of an auditory stimulus. Now let's align things by movement time, which happens later in time in this task. And you find that essentially every region of the brain we've recorded from shows action-related activity, including primary visual cortex, etc., although a bit later, some areas earlier, some areas later, but movement-related activity is all over the brain, okay? I mean, all over the places where we recorded from. And reward-related activity is even more global. So yeah, sure, you'll find it in the places where you expect it, prefrontal cortex and ventrotegmental areas, etc., but it's everywhere, okay? That's not something you're going to get from the textbook, okay? And I would like to now ask, where are the decisions made? This is a big question, right? And so now you'll realize that out of 20,000 neurons, I've shown you 0 neurons. So now I'm going to show you 2 neurons. And specifically, I'm going to show you a neuron in M2 or, you know, prefrontal cortex that clearly has some decision-related activity. Here it is. This is, by the way, the spike waveform of this neuron. You know, imagine that the neuropixels probe has, you know, it has sites that are offset from each other, 20 microns apart. And if this was the actual probe, it would go up to the, you know, the 100th floor. And more, actually. And these are rasters, depending on whether the animal chose right or chose left. And you can see that even before the movement onset, you can tell that this mouse is going to go right or left. And We have also analyzed the activity as a function of stimulus contrast, etc., but I'm going to be quick. But now I'm going to show you a much more impressive neuron that is not in cortex. Guess where it is? It's in the midbrain reticular nucleus. Check out this neuron. Okay, first of all, amazing spike. Second of all, spikes like a bandit. Third of all, incredible distinction between choosing right and choosing left, way before the movement is made. Okay? In fact, this is the earliest place in the brain where we could find movement-related decisions, you know, chosen actions. And maybe some people in the room have been studying the reticular nucleus for years. Certainly Maruzzi and Magoon, you know, there's an incredibly illustrious history of studying this nucleus. It's not like I discovered it. It was in my ignorance that I didn't realize what an astonishingly wonderful place it is. Now I want to use a little model to tell you where we find decision signals. And the model is extremely simple, not very helpful that it's down here. It's a simple linear regression model that says that the response of a neuron in a particular trial at time t is going to be a particular thing that the neuron likes to do, whatever, trial related, plus some function of time with contrast that the animal sees on the left side, some function of time with the contrast that the animal sees on the right, and something having to do with a decision about going left or right, okay? So now we're going to ask, and we're going to cross-validate like maniacs, so the numbers that you will see here are very small, and also we're going to average across neurons, including a ton of neurons that carry no decision information. And what we ask now is, how much do we lose if we ignore this decision term? I hope you can see down here, apologies. I was hoping this would have been a little higher. And so here we go. In the midbrain reticular nucleus and in the deep superior colliculus, you get these early decision signals that are quite strong and quite early. Later, you get them in secondary motor cortex in caudopotamen, and yeah, then that's it. They're nowhere to be found. So I'm going to summarize, if that's it, yeah? So I'm going to summarize with a series of summary slides. So this, I said, is broadly speaking the textbook view of how the brain works, and let's see, well, we found some measurements that seem to agree with it, especially when we were doing wide field imaging and recordings. Also, we found that there is this flow in cortex, which seems to agree with this. When we started recording all over the brain, we found something that looks more like this, with parallel pathways, but that's okay. The textbook does say these kind of things. It does say that the caudopotamen gets input from lots of brain cortical regions. It does say that it projects, not directly to the superior colliculus. This is still broadly consistent with the textbook model. Another thing that we found that was in agreement with that is sensory activity is where the textbook says sensory processing and more sensory processing is in sensory regions, visual, auditory. It is the way the book says. And now there are two ways in which what we found is really not in agreement with what you would get out of the book. The book doesn't literally say, reward processing is only in one place or action execution signals are only in one place, but there's a strong implication. So if we now go look at action signals, we find that they are all over the brain. And if we go look at reward processing signals, we find that they're, again, all over the brain, with different timings, don't get me wrong, but still. So now we think that to understand how this thing works, you do need to get to the single neuron level, because we're finding that different neurons do very different things even in areas that we thought were homogeneous, and I don't have time to explain to you that. Therefore, we need to record from all over the brain. And our lab is funded by the Salmons Foundation, by the Wellcome Trust, to do this work. And the data shown you come from one guy, one postdoc, Nick Steinmetz, who recorded from 20,000 neurons in 5 mice. So this is doable at the level of the single lab, but it's a really complicated problem. Even just acquiring the data, but also making sense of it. This is no longer on the scale of what a single lab can do. And for this reason, we have teamed up with another 20 labs, and we formed the International Brain Laboratory, which is still funded by, again funded by the Simons Foundation and by the Wellcome Trust. And the idea is to do what we did with a slightly different task, maybe more interesting to people interested in value than the one I described. And we think that once we do this in 10 experimental labs and 11 theoretical labs, we're going to do, roughly speaking, 20 times more mice and roughly speaking 25 times more neurons. And the idea is to make the data available to everybody. And I can tell you much more about this, but I want to end here and thank you for your attention.
Excellent. We're actually running on time, so there's plenty of time for questions. Up front over here, I think, first. And then Esther next.
Thank you, great talk. So when you say that, like reward-related activity, like did you mean so it's correlated with the time of reward delivery, or you actually have trials that omitted the reward and compared both situations? And is the scope in the brain that expresses this reward-related activity like kind of aligned with any neuromodulatory projections throughout the brain?
Okay, so the first question is whether, what about the reward? Is it about the sound that the reward thing makes, or is it actually having the reward? Or the way we did the experiment in most mice was just plainly doing the experiment. We didn't just give the reward by itself. So we don't have it for this data set, we have it for other data sets. But in fact, the auditory activity that I showed you, that was with the same sound that is associated with the reward, and so that distinguishes the two. But there's clearly more work to be done. It does happen at the time of the reward, though, the activity throughout the brain. That's all I can say for now.
Esther, and if someone else has a question, put your hands up and we'll retrieve the mic and hand it off.
Okay, I have a statement and a question.
Where are you?
Right here.
Right
Yes, hi.
Hi. Speaking as an author in the Candel textbook, we are now restricted to no more than 4 new figures. Usually you have about 15 to 20 figures in any chapter of the book, so it sort of stayed. The question has to do with the somatosensory effects. When you looked at your mouse on the image doing the task, there was a lot of whisker movement and a lot of hand movement.
Yes.
And if some of those signals that you recorded related to their walking and the whisker movement rather than the decision in the task.
So much. First of all, there's no walking here. There's walking in other tasks, but not in this task. But as you observed, there's whisking, there's all sorts of things. And in fact, we have a whole program of study in which we regress the neural activity that we see on to the face and the whisking and the pupil. And I wish I could put those slides in. I was out of time. But so much of the variance of what happens in somatosensory cortex, in motor cortex, in even primary visual cortex is explained by all that. And while I'm at it, I also want to say that this task that I showed you is not a great task to study motor control, because different mice do it differently. Just like different people drive differently, some people are just like holding the wheel like that. Our mice can do it with 1 paw, with 2 paws. Some mice stop and grab the straw to drink every time they get a reward. Some mice don't. So I bet the motor activity is totally different across mice. And so I would say that this is a very good task up to the motor execution. From then on, I don't know that this is such a great task.
Kenji first, and then Terry.
Hi. Thank you for the impressive result of recording. So regarding the widespread activity, it's kind of related to the previous question, but visual presentation may evoke like eye movement, and also reward delivery provoke like a mouth movement of other motor behaviors. So could you further kind of clean or regress the different activity, neural activity regarding those associated sensory
Yes,
motor
absolutely.
component?
Yeah, absolutely. So we're building regressors that, models that have lots of regressors. And there are some regions, I forgot the name, where licking is incredible as a predictor of neural activity. I forgot the name of the region, I apologize. So no, we're putting all of those regressors in. We're also lucky that our mice vary in the time between the stimulus that we show and the execution. They're fairly consistent in the 350 millisecond range, but there's variation, and this allows us to distinguish these events. Also, we have lots of blank stimuli where there's nothing on the screen and the mouse correctly doesn't move, which takes away a lot of regressors, which is very helpful, or times when there's nothing on the screen and the mouse does move, which again helps us regress. So there's a good combination of regression, and so in that sense, we're not in a bad place. I would say that we would be in a very bad place if we wanted to regress specific body movements, because we don't have sensors on the paws. I cannot guarantee to you that those 5 mice did the task the same way from the motor standpoint. That's where we're weak.
So how much do they move their eyes?
Oh, mice don't move their eyes very much. They move them about once every 3 seconds, as opposed to us, we move them about 3 times a second. And they move them mostly, at least when they're head fixed. It's possible that in nature they do it differently. They move them mostly on the horizontal plane. And they don't have a fovea, so they have no reason to move their eyes towards the stimulus. And we track the eye. In fact, much more interesting than the eye position is the pupil dilation, which predicts a ton of variance.
Okay. So while Terry asks, put your hand up and we'll try and get the mic to you for the next question. In the back, there are a couple of people back there.
So it's really exciting to see so much activity throughout the brain during the motor period and also the reward period. So there's we know there's efferent's copy and corollary discharge signals, which interestingly even get down to the visual system.
Yes.
And that could have some very important feedback information. Regarding the reward, we know that the dopamine neurons project very broadly and diffusely throughout the cortex. So
not directly. Dopamine neurons go mostly to prefrontal cortex, but there's ample possibility for signals to make their way from there back.
But there are other neuromodulatory systems, but basal ganglia is heavily, and even I bet this midbrain reticular nucleus has probably got a lot of dopamine signals.
Yeah. Well, let me elaborate on what you said. Number one, efferent's copy. That's probably what we're measuring. Don't get me wrong. Number one, and number two, maybe the people who do back prop and have error signals go all the way to the front end. They're not that far off. Maybe, you know what I mean? Maybe it's not such a bad view of how the brain works.
In the back over, yeah.
Hi, it's a fantastic talk. I had a question about the test where you inactivate visual cortex and show that it's causally related in the task.
Yeah.
And later on you showed this much larger and earlier activation in the superior colliculus. So I was thinking about blind side patients who can still presumably do very similar tasks. And my question is, do you have strategies to eliminate that possibility whereby your inactivation in visual cortex is not directly interfering the actual information process, which might be happening in subcortical regions?
Yeah, that's a great question. First of all, I think we've done a very careful inactivation study. We're currently inactivating the superior colliculus, so it will be. But it is also becoming increasingly understood that even with optogenetics, when you inactivate transiently, you're changing the system. And if a particular system is used to get 10 spikes per second coming overall from visual cortex, and now suddenly doesn't get them, maybe it just stops doing its job, okay? And so I don't want to overinterpret the results. I think they can be used in a different way, which is, you see this blob of activity that is massive in primary motor cortex and primary somatosensory cortex, which was that yellow blob. I think you would be, I would understand it if you said, well, let me see if that's needed, and I showed you it's not needed. I can shut it down, and the mouse will do the task perfectly. But if I shut down visual cortex, the mouse will not do the task perfectly. Right now, we're using JAWS to inactivate superior colliculus and see what happens in this task. I bet the mouse will be impaired because it's using a combination of superior colliculus and primary visual cortex. in my view.
In the back over here, we have one. Yeah, and anyone else want to put their hand up so that we can avoid Tony getting a question?
Thank you for the really interesting talk. My question
Can you wave your hand? I don't know where you are. Yes, hi.
Hi, thank you. So my question is related to the midbrain reticulous nucleus.
Yes.
So you said there you observe the strongest and earliest choice-related signal, and I wonder if you have any thoughts on what happened next, and why is the later signals even weaker than this earlier?
Sorry, the second part of your question, you said what happens next, and then you said, and why is the?
Why are the later signals even weaker than the earlier strongest decision-related signals?
You know, I wish I had the answers to your questions. They're really good questions. I don't know. The reticular nucleus wasn't even in my mind until we did these experiments. And we now know that a lot of action is driven by the colliculus. The colliculus has this layered structure that goes from vision to action. It's a tectum in birds. And just underneath is the reticular nucleus. That's the core of the brain. That's the part of the brain that does stuff, and including sleep and control of all sorts of things. So maybe in retrospect, it's not so crazy that we found those signals there. Whether those signals are causally related to what the mouse does, I don't know, but it would be a tough experiment because the reticular nucleus, just like the name kind of implies, it doesn't seem to have a lot of structure. The neurons that we find to be interesting live right next to neurons that are not interesting at all. So I don't know if in activation what would happen there. I guess we could play with it.
Okay, and back here.
Yeah, I had a similar question about the mRNA neurons and how to causally sort of establish.
Well, I just said I can't do that.
Yeah, exactly.
Okay.
So, sort of like maybe, do you have any thoughts on whole brain ways to establish causalities? Like, maybe you can talk about the neuropixels opto or.
Okay, so I can tell you that the next generation of NeuroPixels probes is going to have 4 shanks, and also there's going to be a version that does optogenetics, and it's going to have on-site lasers that produce light at different levels. So I suppose that if one found some interesting neurons and one managed to get some channel rhodopsin in there or some optogenetics, one could then activate them or inactivate them. But you know, for us, even just having the data of what happens in the brain where at neuronal level, that's going to keep us busy as a community for a long time before we get to causality, in my view. Because, you know, just think about the correlations. There are 1,000 neurons at a time in the brain of a mouse doing something. When have we had this before, you know?
Okay, it's 12:30. Let's see a show of hands. Who wants to hear Tony's question? Go ahead, Tony.
I don't care. So, Matteo, yesterday you made an observation that in all the time you're in my lab, you only once ever did anything I told you to do.
No, I said that you asked me once and I didn't do it, but anyway.
Yeah, but that's right. You've had a series of questions, right, from different people about different aspects of the activity you showed, and in each case, the activity that you showed was well explained by something which I think you will find in the textbook. You've taken an animal in an extremely complicated task involving vision, motor control, recurrence, reafference, modulation, reward. Mm-hmm. And you have, of course, found activity in all the places in the brain where vision, motor control, recurrence, motor control are supposed to act. So I would put it to you that you actually confirmed the textbook model of brain function because you haven't found any activity that hasn't been explainable in terms of things in the textbook.
Well, okay, so first of all, the task is not that complicated. It's simple enough that even a monkey could do it. And
complicated in the sense of having many components.
Yes. Second of all, I don't get that sense from Kandel and Schwartz or other books that reward signals or motor-related signals would appear throughout the brain. If you think you get that sense, then I'd love to read that book, and I just didn't get that sense. And if the brain does what you think we knew, that's also a good thing. But a lot of this was a surprise to me.
Okay, we reconvene at 2 o'clock, and let's thank Matteo again for a really thought-provoking